Hey guys, welcome back. This is Lei. In the last video, I showed you how to identify peaks in ChIP-seq data using Homer. In this video, I'm going to show you how to visualize these peaks. In ChIP-seq data, we often see beautiful graphs from a genome browser with feature-enriched tracks, or colorful heat maps. So what do they mean, and how do we make them? Let's dive right in. Here I introduce three tools for ChIP-seq data visualization: the Integrative Genomics Viewer, the IGV, the UCSC Genome Browser, and deepTools. The IGV should be your go-to app. It's lightweight, can be installed on your laptop, and has a graphical interface. You can directly visualize your data in IGV without much learning. The UCSC Genome Browser is feature-enriched and highly customizable. You can compare your data with multiple other datasets available online. The deepTools is a comprehensive command line tool that generates multiple graph types as well as file format conversion. The files needed for visualization are PIC files in BED format and coverage files in BigWig format. This is the typical format of a BED file, a TSV file in 3 columns: chromatin, the start points of peaks, the end points of peaks. They're basically the genomic coordinates of the peaks. Here is how we make the BED files. First, we activate the Homer environment we created in the last tutorial. All the essential tools should have already been installed. Next, we fetch the chromatin sizes using fetchChromSizes command. Here we use human genome. You can replace it with MM10 for mouse genome. Next, we convert our PIC file into a BED file using path-to-bed.pl command. The input of this command is a PIC file. The output is a BED file. Next, we sort our BED file first by chromatins, next by coordinates using sort command. The input of sort is BED file. The output is sorted BED file. The LC_all equals C is an environmental variable that keeps consistent sorting. If your BED files are too big, you can compress them into BigBed files using bedToBigBed command. Input our BED file and chromatin sizes file. Output is a BigBed file. We make BigWig files using make_UCSC file command from the Homer package if you caught peaks using Homer. The input of this command is the ChIP sample tag folder from Homer. The output BigWig file will be saved in the same tag file if you specify the output directory as auto. You can choose to normalize your ChIP sample against the input control sample by including the input control tag folder using the -i option. We also need the chromatin sizes file and the style option, ChIP-seq. If you didn't use Homer to call peaks, you can still make BigWig files using BAM files directly with the BAM coverage command from deepTools package. The input of BAM coverage is a BAM file. Output is our BigWig file. Bin size is the resolution. The smaller the size, the higher the resolution. Normalizing RPKM. The genome size is a human genome size, 3 billion base pairs. Number of CPUs, 8. And here we make BigWig files separately for our ChIP sample and input control sample. Now we have BED files and BigWig files. We can visualize them in IGV. Here we have 5 files in total, 4 BigWig files and 1 BED file. Let's import them into IGV. Select the appropriate reference genome. Here we use human genome. And the genomic region we are interested in. Here we use CCND1 as an example. In IGV, each row is a track representing the coverage distribution of a sample. In this figure, the top track is our chip sample, adjusted for the input control. The BigWig file was generated using Homer. The second track is the same, except the BigWig file was generated using deepTools. The third track is the unadjusted chip sample. The fourth one is the input control. You can change the color of the tracks or the height of the tracks by right-clicking on the tracks. And make sure you click on Group Autoscale for the unadjusted tracks. Don't scale the adjusted tracks. Note, not all the bumps are peaks. That's where our beta files comes into handy. The last track is our beta file, which contains the peaks identified by Homer. And these green rectangles are peaks. As you can see, they're nicely overlapped with what we see in the tracks. You can do the same with the UCSC Genome Browser and add a lot more tracks to compare with your own data. However, here's the catch: you can't upload your files to UCSC Genome Browser. The browser only accepts links of your files. That means you have to host your files online somewhere else. And unfortunately, most cloud storage won't work, such as Google Cloud, OneDrive, Dropbox, and Box. The only online cloud storage that works I can find is called Cyverse. It provides a 5 GB free storage. So you can create an account of Cyverse and upload your files to Cyverse and get a link of your files. Once you have the link, go to the browser, select the reference genome, go to My Data, Custom Tracks, and paste your link in this format. Let's look at the structure of the tags. You need a track type. We need the BigWig, a name, and we have a USF2 ChIP-seq as the track name, and the big data URL. This is a URL you get from Cyverse. And you can customize the color. Here's the RGB color, and the view limits is the y-axis over here. It's basically the counts of the peak, the highest counts, and the height of the tracks. And you do the same for each track. And for the peaks, make sure you specify it as the BigBed. And once you finish the text box, submit, and you get the results. Here, the top track of our chip sample, and the second one is input control, and these red blocks are the peaks from our bed file. In RNA-seq data, we use Heatmap to visualize gene expression. Similarly, in ChIP-seq data, we use Heatmap to visualize the strengths of peak signals. In this Heatmap, we stack up all the peaks and rank them by the strength of peak signals. The profile plot shows the same information, except it can include multiple samples for comparison purpose. Here is how we make these plots using deepTools. First step, we have to make a signal strength matrix using compute matrix command. The input is our BigWig file, the beta file. The output is the compressed signal strength matrix. Dash B is the region before the center of the peaks. Dash A is the region after the center of the peaks. Bin size is the resolution. Number of processors is the CPUs you want to use. Once we have the matrix, we can plot the heatmap using plot heatmap command. Input is our matrix from the above step. Output is the image file. You can specify color and resolution. and all other options are not important. They are just names of the profile. For the profile plot, you can make it using plot profile. Input is the same matrix. Output is the image file. And you can specify the colors, names, and the resolution of the image. We can also visualize peak signals around TSS across the whole genome in the same way with 2 extra steps. First, download a gene annotation file. Second, extract TSS information from the gene annotation file. You don't have to understand this complex command. You just need to know the output of this command is a BED file that contains TSS information. Once we have this BED file, we can compute the matrix using compute matrix command in the same way. Input is our BigWig file, and the TSS BED file. Output is a TSS matrix. And then we plot the heat map using plot heat map command. Input is a matrix. Output is a heat map image file, like this one. And this is all we have for today's tutorial. If you like it, subscribe, and I'll see you in the next video.